السلام عليكم أعزائي الطلاب. This is the second lecture regarding biological membrane or cell membrane. We discussed in the first lecture the basic structure of the cell membrane, and we knew that it consists mainly of a bilayer of phospholipid embedded in its proteins, cholesterol, and glycogen. So let's speak a few notes about the permeability of the cell membrane. It has the highest permeability for lipid-soluble compounds. That means for hydrophobic compound. Example, steroids. Because it is a hydrophobic barrier, the highest permeability of it is for lipid-soluble compound, for lipophilic compound. We mean by this compound, for example, the steroids can enter and exit the cell membrane easily. Also, O2, CO2, and H2O, a small compound, uncharged compound, can pass the membrane easily. And this is crucial to maintain the respiration of the cell. We need to obtain oxygen and to get rid of CO2. And H2O, also it is crucial to let in and out easily and freely in order to maintain the tonicity of the cell, or what's called the osmotic pressure of the cell. On the contrary, it has poor permeability for ions, which are small charged molecules and polar compounds. For if the molecule have a charge, it would be repelled by this hydrophobic barrier and cannot pass the membrane easily. This line represents the permeability coefficient of the cell membrane. To the right, it is the highest permeability coefficient, so the membrane is highest permeable here, while here it is the lowest permeability. We see water have the highest permeability coefficient. That means the water can enter and exit the cell membrane nearly freely, while on the contrary, ions, which are small charged molecules such as sodium, potassium, and chloride, cannot enter and exit the cell membrane easily because the membrane have the lowest permeability coefficient for them. So there should be present transporters, channels, some way for exchange of these important ions. So here comes the functions of proteins. The proteins in the cell membrane have a lot of important and crucial functions. First function is as a support. Proteins may be part of the cytoskeleton of the cell. They help to maintain the shape of the cell, the configuration of the cell, the integrity of the cell membrane. All of this may be attributed to the presence of proteins. As channels and also as the transporters, both of them help in the transport of charged molecules, example ions, across the cell membrane. What's the difference? Channels act passively, so they are only as a corridor through which the ions pass the hydrophobic barrier, while transporter actively transport these ions, usually may require energy or by help of other ions. Cell surface identity. We said before that the glycocalyx is the component in the cell membrane responsible for the identity of the cell. So why here we mention this? Because the glycocalyx mainly consists of the carbohydrate part of glycoproteins and the glycolipids. And since the glycoproteins are more abundant in the cell membrane, so the cell surface identity may be attributed to the proteins. And we mean by this the glycoproteins. Adhesion. Help the proteins for adhesion of cells one to each other. Maybe act as receptors for hormones and the neurotransmitters. These effectors or effective substances act on the cell membrane through attachment to receptors made mainly of proteins. And as enzymes, cell membrane may act as enzymes whether on the external leaflet or in the internal leaflet. These enzymes are responsible. In general, proteins can be divided into two parts: integral proteins and peripheral proteins. So we have two types of proteins, and there are several differences between integral proteins and peripheral proteins. You are usually asked about these differences. You are asked to differentiate between integral proteins and peripheral proteins. And it is easy to remember these if you understand few points. Let us speak about integral protein. The main point in integral protein is that they contain large numbers of non-polar, we mean hydrophobic amino acids. You know, amino acids can be classified into polar amino acids, which are either acidic or basic or uncharged amino acid. Acidic, for example, glutamic acid, aspartic acid. Basic, for example, arginine, lysine, histidine. Polar uncharged, for example, threonine and others. And some non-polar amino acid, for example, leucine, valine, alanine, etc. So the polar amino acids are here is less than present in the peripheral proteins. The integral proteins contain a higher percentage or large quantity of non-polar, of hydrophobic amino acids. Since these integral proteins have some hydrophobic properties, that is why they interact extensively, this is a very important word, extensively, with the hydrocarbon side chains of fatty acid. Since they contain hydrophobic part or hydrophobic portion, this hydrophobic part make them interact extensively, strongly with the hydrocarbon side chain of the fatty acid. Now, since they interact extensively, they are difficult to be separated. So separation of integral proteins، من كلمة integral أيضا تعني جزء أساسي، فالintegral عادة من الصعوبة فصله. So they contain hydrophobic amino acid, enable them to interact extensively with the tails of the phospholipid, with the hydrocarbon side chains of the fatty acids and the phospholipid, and making them difficult to be separated from the. And since they interact with the hydrophobic part of the membrane, they generally span the cell membrane. Generally, not always. Not all integral proteins span the cell membrane. Some of them span the cell membrane even 7 times, and others do not span the cell membrane. But generally, integral proteins can span the cell membrane because they contain hydrophobic part. And since they span the cell membrane, they can act mainly as channels, as transporters, because both of them requires the protein to cross the cell membrane or to span the cell membrane. They may act as receptors also, or as structural proteins, but mainly as channels and transporters. Example for integral proteins is G protein. This is an important receptor, especially for insulin, and it helps to transport the hormonal signal from outside of the cell to the inside of the cell. Now we'll come to the peripheral proteins. In the contrary, peripheral proteins, they contain merely polar amino acids. So they do not contain merely non-polar, they contain merely polar amino acids. That is why they are attached to the polar ends of phospholipid. They cannot interact with the hydrocarbon side chains. They cannot interact with the tails of phospholipid. They interact with the polar ends because they are polar. They attach to the polar ends of the phospholipid or to the integral proteins, because the integral proteins also have hydrophilic part by weak electrostatic bonds. This is not a hydrophobic interaction. This is a polar attachment which is weak electrostatic bonds. And since it is weak, it is easily separated by ionic solvent. They can be easily separated by ionic solvents. And since they do not contain mainly hydrophobic part, they cannot span the cell membrane. Peripheral proteins do not span the cell membrane because they cannot span the hydrophobic part. So they function mainly as mechanical support if they are present mainly in the inside, they are part of the cytoskeleton, or to regulate hormonal activity. How regulate hormonal activity? Since they may be attaching to the head or to the polar part of integral proteins, they receive the signal from the hormones and they transport the signal to the integral proteins. So act as a bridge to regulate hormonal activity in conjugation with the action of integral proteins. Example of them is incretin. So here, this is the proteins. We see this is protein spanning the cell membrane, so this is an integral protein. This is also integral protein because they enter between the hydrophobic side chains, but it doesn't span the cell membrane. So not all proteins spanning, not all integral proteins span the cell membrane. And this helix here is usually the hydrophobic part of the integral protein. On the other hand, these are peripheral proteins. They are present either to the external or to the internal part of the cell membrane. They are here attached to the integral proteins. They may be attached to the glycolipids or to other parts. And they are usually peripheral proteins, do not span the cell membrane. Here, this peripheral protein may act as a receiver of hormonal action. So here, this peripheral protein and this may act as a mechanical support. To understand the function of proteins present in the cell membrane more, we'll have an example of the membrane proteins. We'll take an example of the proteins present in the RBC, the red blood cell, responsible for the respiration of whole of our body. Here we see these are the proteins present in the cell membrane of RBC. We have two important integral proteins, the first called band 3 and the second called glycophorin. You see, both of them are integral proteins. Why? Because they span the cell membrane. This spans the cell membrane twice, and this may be more than 7 times, more than 8 times. So it spans the cell membrane. And these are peripheral proteins present inside of the RBCs: ankyrin, spectrin, actin, and the protein 4.1. This usually do not even included within the membranes of the cell membrane. It is attached to one of them because it doesn't have a direct action on the cell membrane. So we have two integral proteins, band 3 glycophorin, and we have 3 peripheral proteins, ankyrin, spectrin, and actin. What's the functions of these? Let's take first the band 3. Band 3 is an integral protein, act as a bicarbonate chloride exchange transporter. So it acts as a transporter. Why this is important? Since the RBC, one of the functions of the RBC is to carry oxygen and CO2. Oxygen is easily incorporated inside hemoglobin, so it can be carried out easily. While CO2, to be carried out, it should be converted first into bicarbonate. So in the peripheral tissues, RBCs are loaded with bicarbonate, and it takes it to the lung where it is exported to the alveolar air space to be exchanged. So in order to maintain a high concentration of bicarbonate inside the RBC, band 3 is a very important transporter to transport bicarbonate in exchange of chloride, and so accumulate bicarbonate inside the RBC and then let it discharge or excrete it in the capillaries of the lung. So this is very important function of an integral protein presence in the RBC. The glycophorin have less important function, still it is crucial to the action of the RBC. It provides an external negative charge that repel other cells. We see One of the most important functions of the cell membrane is net charge stabilization. Glycophorin is the protein responsible for this. It maintains a negative charge outside of the cell membrane so the RBC do not stack to each other. If the RBCs stack to each other, they cannot pass the small capillaries, especially present in the spleen, and they will be destroyed and there will be hemolysis. So this glycophorin helps to maintain a negative charge on the RBCs so they repel each other and they do not stack and they can transport easily inside our plasma. We see both of them are very important integral proteins present in the RBC. What about the peripheral proteins? Ankyrin, actin, and spectrin are very important peripheral proteins. They comprise the inner membrane skeleton. They form the cytoskeleton of the RBC. So, this is band 3, this is the glycophorin, and these three, actin, spectrin, and ankyrin, comprise the cytoskeleton of these three proteins maintain the RBC in this biconcave shape. So this biconcave shape is maintained by the action of the cytoskeleton of the RBC, which consists of the ankyrin, spectrin, and actin proteins. In a certain condition called hereditary spherocytosis, there will be defects, usually of ankyrin and maybe of spectrin. When there's defect in these proteins, the cytoskeleton will not be very effective, and so the RBC, rather than being biconcave, it will take the spherical shape. That is why it's called spherocyte. So rather than this biconcave shape, it takes a spherical shape. And this spherical shape cannot be squeezed inside the small capillaries in the spleen, and so these RBCs will have a shorter lifespan and will be destroyed. So in hereditary spherocytosis, it is one cause of hemolytic anemia. What that means, there will be a shorter lifespan of the RBCs because they are hemolyzed when they pass inside the spleen, and the patient will have a decrease in the RBC and decrease in the hemoglobin and a condition called anemia. So one of the causes of anemia is hereditary spherocytosis, in which there will be conversion of the RBC from biconcave into spherical shape because of defect in ankyrin of spectrin proteins that maintaining the cytoskeleton of the cell. We see all of peripheral proteins and integral proteins are very important to the action of RBC and all other proteins. This is me, and thank you for your listening.